Hi, in this protocol, we're going to look at converting messenger RNA to complementary DNA. You typically want to do this because DNA is way more stable than the RNA that you've extracted from the cells. There are multiple approaches for enriching for your messenger RNA. You may use Oligo-dT segments or nucleotides that are used as primers, and in this case they bind to the adenines that are at the end of a messenger RNA transcript. And in this case, when you go on to make your complementary DNA, you will use primers that target these oligo-DTs, and it's a preferred form of creating your cDNA because you actually end up having all your cDNA copies terminating at the 3' end of the messenger RNA, and it also produces the longest contiguous complementary DNA fragments. When using an oligo-DT approach, so you have your short thymine nucleotides, and these ones can be any length. So in this case, it's 23. And what you do is that it forces the primer to anneal to the start of the poly-A tail. So they typically anchor oligo-DT primers, and in this case, it forces the primer to anneal to the start of the poly-A tail. And it prevents priming at internal sites in the poly-A tail. So by providing these oligo-DTs, you then are able to use oligo-DT primers, and in this case, the synthesis of complementary DNA will occur from the poly-A tail that is targeted. However, not all applications would choose to use an oligo-DT approach for messenger RNA enrichment. So in instances where you had your ribosomal RNAs or other messenger RNAs that you're not trying to study. So for instance, if you want to get rid of messenger RNAs that are related to blood, for instance, if the cells came from a cell culture where you've used blood in the process, or you may want to get rid of ribosomal RNA that's related to mitochondria because you're not studying that. So in that case, you can just target those ribosomal RNA and other RNA pieces that you're not interested in. And in that case, you should be left with messenger RNA, but it's not, you didn't use an oligo-DT based approach to enrich. So in such instances, when it's time to make your complementary DNA, the primers that you're going to use will not be primers that are targeting just oligo-DT. You'll use primers that are called random hexamers. So here's a schematic of what it would look like, and this is from TipTop Bio. Credit to TipTop Bio for creating this schematic. So where you're using random hexamers, they will just target different portions of the gene body, as well as in some instances there will be a mix, so they will also target the poly-A tail. But what will happen is, because you don't have only the poly-A tail being targeted, you get different fragments of products potentially. But where you have the oligo-DT, you're only targeting the poly-A tail, and you can get longer stretches of the gene. You may also want to target a specific gene, and so you just give that primer at that complementary DNA stage. No need to put hexamers, random hexamers, or oligo-DT primers. So as mentioned, typically your random hexamer mix is a mix of hexamers as well as oligo-DT stretches of thymine oligos. And so in this case, you get covering of the entire RNA templates, including your messenger RNAs and some non-messenger RNA sequences, polyadenylated RNAs. So from looking at this schematic, what you can see then is you are likely to get shorter fragments of amplification or copying where you're using the random hexamer approach. The products here can be used for detection of short reverse transcription based PCR products. So let's walk through a typical protocol. So let's say if you're using an Oligo-dT based primer because you enriched based on Oligo-dT, then you may need a particular concentration of starting primer versus if you use a random hexamer based approach. And so in the protocol I'm using, it requested 5 micromolar if you're using an oligo primer, whereas Oligo-dT primer. But if you're using a random hexamer primer, then you needed 6 micromolar as your starting concentrate or as your final concentration. If you are using a gene-specific primer, then typically you're looking at 0.1 to 1 micromolar, so much less concentration required since you're only targeting that one gene. Quite a straightforward protocol. The first step is we're going to denature both the RNA and the primer mix. And how we do this is to use a thermocycler as well as some reaction mixes, PCR machine. The reaction volume in this particular protocol is 8 microliters. And so this means that everything needs to add up to 8 microliters. So what's required is 2 microliters of your primer, whether it's an Oligo-dT based or random hexamer based. So you need 2 microliters. And that of course means that you've got 6 microliters to play with. So your 6 microliters can be taken up by the RNA that you're wanting to synthesize to do the reverse transcription on. So you can use up to a microgram of it in 6 microliters, or if your RNA is super concentrated, then just make up the volume to 8 microliters once you've added your RNA. The first step is to heat your reaction mix at up to 65 degrees Celsius, and this time for just 5 minutes. The tube should be briefly spun and then placed on ice immediately. Now we're ready for the reverse transcription step. This is where we're going to use an RNA-dependent polymerase to copy the RNA template into DNA. The reaction volume here is 10 microliters. And in my particular protocol, the reaction mix comes as 2 times concentrated, while the enzyme mix, which will have the reverse transcriptase, is 10 times concentrated. So I need just 2 microliters of it. So the reverse transcriptase can be sourced from multiple sources, I imagine. So in this case, in the kit that I have, the reverse transcriptase is from Maloney-Murine leukemia virus, which is abbreviated on the screen. So how this particular reverse transcriptase is obtained is that the murine reverse transcriptase, Maloney-Murine leukemia virus reverse transcriptase, is expressed in an E. coli vector, and apparently it results in a product where 16 additional amino acids are at the N terminus and 13 amino acids are at the C terminus, and the construct results in a fully functional reverse transcriptase. And it also has functional RNase H domain. So to check that the reaction is specific, we also do a negative control. So this is where we've got the RNA, we've got the reaction mix, but we don't put any Maloney reverse transcriptase, Maloney-Murine leukemia virus reverse transcriptase. We leave that one out so that we can check that our reaction is specific. And so here we need to heat it up to 42 degrees, and we're going to incubate it at 42 degrees for a whole hour, for 60 minutes. Once that's done, we want to inactivate the enzyme, the reverse transcriptase. So we go ahead and place it at 80 degrees Celsius for 5 minutes in order to inactivate the reverse transcriptase. And voila, the complementary DNA is now ready. If you're not ready for PCR, you can pop it at minus 20 degrees Celsius until you're ready. So this product can now be used in PCR reactions.